Dr. Sonia Padwal is a researcher at Hamilton Centre for Kidney Research, Firestone Institute for Respiratory Health, and the McMaster Immunology Research Centre. She completed her PhD in 2017 in medical sciences at McMaster University. Her doctoral work investigated the complications of long-term peritoneal dialysis for patients with chronic kidney disease. She is now working with the Ask Lab group investigating the mechanisms behind the development of interstitial lung disease, specifically idiopathic pulmonary fibrosis, and also continuing to do research in peritoneal fibrosis.
I'd like to welcome everyone to this Demystifying Medicine seminar. Can you guys hear me? Well, okay. So today I just hope to shed a little bit of light on the PhD, specifically doing a PhD in the medical sciences, and really I want to touch upon the versatility of a PhD. And so I'm just going to briefly go over the objectives as you've seen from the poster, which you should have read. It's been circulating for like a week. So I want you guys to really get an understanding of medical investigation. So this is all the way from basic biomedical research to translational and clinical research. And then also I want to talk a little bit about my doctoral research experience, as well as the wide range of opportunities that exist post-doctorate. So my journey began 10 years ago, actually over a decade ago, in 2008. As a first-year student in McMaster University, I was a first-year life science student. And I completed life sciences. I started my master's in med-sci and completed my PhD, and then now I'm working at Mac. So I just want to get an idea of how many of you started in life sciences or are currently in life sciences or science related. So yeah, basically everyone in the class. And so the reason I ask you that is because when I started off, I had this idea or plan in my head, and I like to call this the life sciences blueprint. And basically what it was my plan. So I was going to start my program in life sciences at McMaster. I would choose my major in first year. I majored in life science honors, complete 2, 3, 4, and complete my 4-year honors degree, apply and get into medical school, and become a doctor. So I'm sure a lot of you can relate to this plan. But what happened was in my second or third year, I had to take a bunch of required courses for the program. And one of the courses was Life Science 2 AO3, Research Methods in Life Sciences. And it was actually the first year it was offered, was in my year, just like ages ago. And what it was taking an in-depth look at the scientific method and application of the scientific method. So basically, it took selected research problems and used that as a vehicle to help students learn about the scientific method. So it went all the way from hypothesis formulation, testing, and communication in the life sciences. And I'm sure how many of you have taken this course? Okay, so maybe half. And so I really got interested in research. And so I started taking more courses with lab components. I contemplated, should I do an undergraduate thesis? I actually didn't end up doing an undergraduate thesis, but because I decided I wanted to actually just do some volunteer work in a lab. And after struggling to find someone to take me, I found this random ecology lab, which was doing research in communities, so ecological rock pool communities, and how these communities assemble. And my job was to count, sort, and categorize whatever this is. So these are zooplankton species. Specifically, they're called Daphnia. And so after this, I was at a fork or a crossroad in the road. And I had to decide whether I wanted to still do medical school or did I want to pursue a master's. And I knew at some point I definitely did want to incorporate research into what I was doing. And so I reevaluated the blueprint, and I decided not to apply to medical school. I put that on hold. And instead, I started a master's in medical sciences, and I wanted to see what kind of opportunities that would lead to. So here I was, starting my master's of science, and I started my master's under the supervision of Dr. Peter Margetts. He's a nephrologist, a clinician scientist at Hamilton Centre for Kidney Research. If you can remember, a few weeks ago, he did the talk on scleroderma and systemic sclerosis. And here's just a look at some of the facilities we have, our lab facilities and some of the conference rooms we have. And my research was focused on the complications associated with peritoneal dialysis. And so I'm not going to get too much into the details, but I just want you guys to understand what type of research I was doing and just get the overall picture. So I was focused on peritoneal dialysis, which is a type of renal replacement therapy for patients with chronic kidney disease and end-stage renal disease. And basically over time what happens is, before I get into what happens over time, let me just explain a little bit about what the therapy is and how it works. So peritoneal dialysis. So the peritoneum is a blood vessel-rich membrane which outlines the abdominal cavity. And because it's so blood vessel-rich, that makes it a great membrane to use for filtration. So here's a schematic where we have a breakdown of how the therapy works. So essentially, patients that are on peritoneal dialysis have a catheter that's permanently and surgically inserted into the abdomen. And through that catheter, they have a bag that's attached, which is filled with dialysis solution, or PD solution. And this solution is really unique because it contains an osmotic agent, which creates a concentration gradient that allows for filtration to occur. So through the process of diffusion, water and solutes can be removed from the blood, and you have filtration of the blood. So at the end, they drain out the fluid, and they can start the process again. So now getting more into what I mentioned, the changes that happen with patients that are on long-term peritoneal dialysis, which is what my research was more focused on. So here is a diagram where you can actually see the catheter is inserted into the abdominal cavity, and you can see the bag containing dialysis fluid is hooked up, and so it'll enter that way. And here we have histology, and this is taken from an animal model of peritoneal membrane injury. So this demonstrates the normal structure of the peritoneal membrane. So you can see three distinct layers. The top layer is a layer of epithelial cells called mesothelial cells. Underlying that is a submesothelium, and then underlying that are the anterior abdominal muscles. So what happens with patients that are on PD for long periods of time is that this structure that I just outlined, it begins to change. So this was a study that looked at 212 biopsies from 130 PD patients. And what they saw was that there was an increase in the thickness of that submesothelial zone. And there was also an increase in the number of blood vessels. And so you can see that more clearly here in our animal model of peritoneal membrane injury. You can see over time, when the peritoneum is injured, you see that thickness in the submesothelial zone. And that thickness is a process called fibrosis, which is essentially scarring due to bioincompatibility of the dialysis fluid. And you also have an increase in blood vessels, which is a process called angiogenesis. So I was studying both of these two elements of peritoneal membrane injury, and I was using an animal model to do this. So it's well known, one of the primary cytokines that initiates this whole injury response is transforming growth factor beta, or TGF-beta. And so we can actually overexpress this in the animals, and we can recapitulate peritoneal membrane injury and study it. And so that's exactly that was the model that I was using throughout my master's and my PhD. And so the reason that these structural changes are so important is because they're actually associated with functional changes. So the actual functioning of the therapy begins to decline over time because we have this thickening, because we have this angiogenesis that develops. And so eventually patients have to be either switched to hemodialysis or transplanted. So they have to be taken off of PD. So the main function of my research was to determine how we can better improve this therapy and figure out the mechanism that causes these complications. And so here's a little cute figure of me in my master's. And yeah, it was a little scary, but I got through it. So at the end of my first year, this was me. And I was, again, at this crossroad where medical school was at the back of my head, but I also had the option to transfer into a PhD. So what I did was I actually applied to medical school that year, and I also transferred to a PhD, and I had the option that if I got any interviews, I could defend my master's, but if I didn't, I would continue with my PhD. And I think you guys can guess I didn't get any interviews, so I continued with my PhD, which was an amazing experience. So I'm just going to get into talking about how that went. So as you get more and more committed to your research project and more immersed into your focused area of interest in the disease you're studying, in biomedical research, it becomes very cellular and molecular based. So this is a complicated diagram of some of the stuff I studied, and I focused actually on the angiogenesis component of peritoneal membrane injury. So remember I mentioned there was two parts, fibrosis and angiogenesis. So I still did a little bit of fibrosis stuff, but a lot of my stuff focused on angiogenesis. And the cell that I was particularly looking at is this little guy right here, which is called the myofibroblast. And I won't get too much into the details, but the myofibroblast is considered the key effector cell in the process of fibrosis, and it also secretes a lot of angiogenic factors. And so as I continued on, I was actually even more focused into one particular protein in this entire pathway, and this was matrix metalloproteinase 9. And we got into this because it had been shown to have an angiogenic role, and also it had been shown to clear basement membrane, which is one of the processes that's involved indirectly in fibrosis. And as I went along with my research in MMP9's role, I sort of bumped into this pathway called the Wnt beta-catenin signaling pathway, and it interacted with MMP9 in some way. And the latter half of my PhD was really focused on Wnt beta-catenin signaling and its role in the entire development of peritoneal membrane injury. And so I'm just going to briefly talk about the different aspects of research. So during my PhD, the research was broken into 3 different types, I guess you could say. So I did a lot of basic science research, and then I did some translational research, and then I also had some clinical research experience as well. So this was my first project. I was looking at the role of MMP9 in peritoneal membrane injury, and this is just a snippet of the beginning of the project. So it was just a study that was done on overexpressing MMP9 so that we could define what MMP9 was really doing in the entire model. And as I mentioned, it's involved in angiogenesis, so we hypothesized that it would have some type of effect on the blood vessels. So when we overexpressed it, we in fact did see that there was an increase in vessel density. And this is really where you're trying to study the biological process in order to create a foundation for applied science. So it's more understanding the mechanisms behind disease development before we can get more into the advanced stages where we're actually applying this to the bedside. And so here's just another complement to that study where we did a knockout model, and we looked at similar outcomes. And so as I was doing this, simultaneously, I had an opportunity to do some translational research as well. So this was really cool. We got patient samples from patients that were actually on peritoneal dialysis. So we would receive these bags of PD fluid, which was essentially from a patient who had had sat through the dwell, and then had all the exchanges, and then they gave us the sample. And so what happens is during this time, a lot of these cells in the peritoneum, they kind of slough off into the fluid, which is really cool because we can then take this fluid, centrifuge it, and we can actually culture these cells. So you can see that right here. And we can measure all sorts of things in these cells. And so because my project was focused on MMP9, we started off by looking at a lot of MMP9 and MMP-related genes. And we actually did find something really cool where MMP9 was associated with some of the functional changes in patients. So we were able to take this and correlate it to clinical characteristics of the patient, which is a lot more translatable. So that was really cool. And then in the last half of my PhD, I got a really exciting opportunity to lead an observational clinical trial. And this was a trial that was looking at the epigenetic determinants of peritoneal fibrosis. So epigenetics essentially is talking about changes in DNA methylation patterns, so gene silencing or gene turning on based on methylation status. And so what I did was I was enrolling patients that were just beginning PD, and then I would do a follow-up visit. So I'd get a sample when they're enrolling, and I do a follow-up visit at 12 months. And so in that 1-year time point, I wanted to see if there were any changes to the DNA methylation pattern. And so similar to the other study, I could grow up the cells and actually look at the DNA from baseline to 1 year. And this was really cool because I actually got to start right at the beginning of the trial all the way till the end. So I started with ethics approval. I was involved in designing the study and going over eligibility criteria. I was communicating with healthcare staff. I was arranging research visits, and I was involved in patient recruitment, which is my favorite part because I actually got to interact with patients. I got to go over the entire study protocol. I got to chat with them a little bit, and I obtained consent if they wanted to participate in the study, and then I got to actually process the sample, and now we're actually analyzing the data from this particular study. And apart from all the research, just want to lead into my next little bit, where I think one of the most important things about research is the actual knowledge translation aspect. And I think every type of graduate work has some type of knowledge translation aspect. And this is more than just publishing in a journal article. This is actually going to a conference or going to a seminar, just like I'm here today, and actually sharing your findings with other researchers and gaining feedback, so gaining insight into gaining their perspectives and gaining new ideas so that you can actually redefine your question. You can branch out into different areas, and you can rethink and reevaluate your own research. And also you can confirm your findings if a lot of other groups are doing similar types of research, and you can build on that and create future directions. So communication and collaborating at these conferences is really important, and it was really a fun experience as well. I got to visit a lot of different places. There's Montreal, Chicago. This was Spain, one of the best conferences. And then on that note of knowledge dissemination, in a similar avenue, I got to do some teaching. So most graduate students get an opportunity to take on a teaching assistantship. And I was TAing anatomy and physiology for first year health sciences. And that was really cool. So I got to lead the lab component and the tutorial component. And I got to develop some skills in education. And I found that I was actually very passionate about teaching, and I really enjoyed teaching. And so I got to improve a lot of my soft skills, and then also had opportunities to teach in the lab, and that was like co-supervising some projects or supervising some undergraduate students. And it also helped me reinforce my own learning because I think teaching and learning, it's just a cycle. So if you're teaching, you're just learning more. So it was a really good experience. And I think with everything in life, you can't be too focused on things. You have to always have a balance. So throughout graduate school, it's very easy to get narrowed in on your project and forget about everything else around you. And so similar experience with me in my first and second year, I was very focused, and I came across this opportunity where there was an amateur art exhibit where students could actually display their art. So this was led by a humanities student in the first year. And this actually helped me learn how to create a balance, and it helped me become more integrated into the graduate community outside of my research domain. And what was really cool was I actually took over this event. So for 2 years, I led this event alongside one of my colleagues, and it was called the Gallery of Graduate Arts. And the whole goal behind it was to motivate graduate students to go beyond their research domain and complement their graduate experience with pursuing their creativity and artistic talents. And this was really cool because we had a whole gallery and we had a whole setting where students could actually come, exchange ideas, and they could really gain confidence in their artistic abilities. And so at the end of it, I did manage to get 3 publications, still working on the 4th one 2 years later. It's in revisions. Hopefully it'll come out soon. And then you have to write this long thing. It's called a thesis, 300-page thesis. Okay, I know, I can't believe it. Okay, so here I am, PhD, a little bit wiser, I hope. Okay, and very quickly, just want to go over the main take-home message from the first part of my talk is the versatility of a PhD. In retrospect, reflecting on my entire experience, you gain so much, like in terms of skills, research skills, transferable skills, but also a lot of personal development and personal growth. And for research skills, like those are pretty obvious, learning how to define a research question, study design, creating a model to actually study the disease, designing experiments, all the way till the technical skills of collecting data, to communicating your understanding, critically analyzing your results and making those connections. So those are all the research skills. But mainly the transferable skills is what I really want to touch upon. So that's more the soft skills that can be applied to other things in life. So communication, so this is talking, being able to communicate to other researchers, being able to communicate to the public, being able to work in a research team, so those interpersonal skills, and being able to collaborate with other researchers. Problem solving under pressure, that's a big one. Another big one is responding to positive and negative feedback. So over the course of graduate work, you always have committee meetings, you always have lab meetings, you're getting feedback constantly from your colleagues, from your supervisor, and it's really important that you kind of learn how to respond to that positive and negative feedback in a way to allow for growth in your project and growth personally as well. A lot of project management, which I've already been over. The biggest one, I think, is adaptability, and it's the most important one. So because you take on so many of these different roles, you become a very adaptable person. So anytime there's some type of changing circumstance, not only in your project, but in other roles as well, you have this ability to adapt and mold to that circumstance. And in terms of personal development, I learned a lot of self-management. Being able to work effectively independently was a big one as well. And also confidence to come up here and talk to you guys, and also creating that work ethic and endurance. And I think one of the most important one is self-reflecting. So again, getting back to reflecting on yourself and highlighting strengths and weaknesses so that you can really build on yourself as a person beyond academics. So yeah, my PhD experience was amazing. So I'm going to talk to you a little bit about my postdoctoral experience, which is also going very well. So wait, hope you guys see, that's what a postdoc looks like. Yeah, so, okay, so currently I'm working with the Ask Lab group, and you know Dr. Ask, he's the head of the whole course. And so I'm having a great time. I'm studying, I've moved a little bit into the lung now, so we study interstitial lung disease, specifically focused on idiopathic pulmonary fibrosis, which is similar. It's the scarring of the lungs, idiopathic meaning no known cause, and eventually results in a decline in lung function. And here we have some characteristic images of how IPF is diagnosed. And at the beginning, when I first started, I did a little bit of research where I focused in on a different cell type. So if you remember, I was more focused on the myofibroblast in my PhD research. Now I moved into the macrophage, which interacts with the fibroblast, so it's still in the same area. And I was focused on something called macrophage polarization, which is, it's a process where the macrophage can actually change phenotypes based on the environmental or biological setting that it's in. And so I was focusing on this process in lung fibrosis. So I did a little bit of studies at the beginning with fibroblasts and macrophages, and I did some stuff with endoplasmic reticulum stress, which is also an ongoing area of interest in our lab. And here's just an example of one of the publications from this year. And as I started working more, I got into more of a project manager type of role where I was managing several different projects. Specifically, I was involved in a lot of gene expression analysis studies in many different projects. And this was really cool because it created this industry link. So we collaborated with many different pharmaceutical partners, as you can see listed here. And it really allows you to create more connections and create that link to industry so that you can transition into that as well. And then right now, we're focusing more on developing a clinical research platform to understand cellular and molecular mechanisms that are involved in fibrotic lung diseases. So I know this is a really busy figure, but what you need to really get out of this is that we are able to take clinical specimens, so both prospective and cross-sectional archive specimens, and we're able to actually use these methodologies to investigate different processes in the realm of interstitial lung disease. And we are combining both clinical and research expertise to do this. And this platform, we're really trying to refine because we want other groups that are studying other diseases to be able to apply this to their area of interest. And so this is more, again, getting more into the translational aspect of research. And we have this entire core facility that is trying to do exactly what I'm talking about. So it's the Molecular Phenotyping and Imaging Core Facility, and it's basically providing these molecular phenotyping services in all different types of research. And we're able to look at these clinical specimens. And so here's just an example of, this is IPF lung tissue taken from a patient, and some of the stuff we can do, we can actually look at different genes. And so this is gene expression. I don't want to get too much into this. But alongside all of this, I also started to get my foot in the door with medical illustrations. So this figure I actually designed. And this is really cool because, as I mentioned earlier, I'm an artist, so this was sort of a nice blend between art and science. And so it's just an example of how easy, how basic something can really be, and take these parts and becomes this block of tissue. So this is just another example of one of the figures that I'm currently working on. It's very preliminary. And lastly, you guys know that I'm involved in education, so I'm TAing Core 2 in this course. And like and subscribe our YouTube channel, to our YouTube channel if you haven't already done so. And there's also Facebook and Instagram. Yeah, so advertisements aside, this is a really cool course because, as you guys know, it's all about knowledge translation. And I love knowledge translation because you really learn so much when you're taking something so complex and you're turning it into a simplified version and you're translating it to the public. And I also love how this course is very student-directed. It's one of the unique courses that's taking, moving away from traditional lecture, which I think really accommodates different learning styles. So in summary, I know I'm a little bit over time, just reiterating the take-home points. So medical investigation involves all types of different research, from basic science, translational, all the way to clinical, and you really do gain a lot of adaptability and versatility with doing a PhD, from beyond academic research skills to transferable and personal development. And there are tons of postdoctoral opportunities, which are also touch upon how versatile the degree is. And my contact info is there if you ever want to contact me. And lastly, I have the 10-year challenge because I've been here for so long. So don't look at that. And I want to leave you with this quote. And that's all. Thank you. I don't know if we have time for questions.